Today, we are undertaking a mission that's truly foundational.
It really is.
We're plunging right into the molecular and, the structural origins of the human axial skeleton.
Mhm.
We're talking about everything that gives us our back, our ribs, and the deep sort of intrinsic engines of movement that hold us upright.
But our goal, as always, is to sort of pull out the critical nuggets, to give you a complete structured understanding of how our highly flexible yet incredibly strong spinal column is built from, well, from scratch.
And the centerpiece of this entire deep dive,
Yeah.
it's this tiny transient structure. It only exists in the early embryo, but it dictates the blueprint for nearly a third of your adult body.
Yeah.
We are tracking the life cycle of the somite.
The somite, it's the foundational segmented unit. I mean, when you look at an adult spine, you see that segmentation immediately, don't you?
Oh, absolutely. The repeating pattern.
The repeating pattern of vertebrae, alternating with the shock absorbers, the intervertebral discs. That segmented body plan it's really the defining characteristic of vertebrates.
And it's what allows for movement, right?
It's the primary mechanism that allows for complex locomotion. So what we're going to do today is track the entire chain of events that takes a simple ball of cells and transforms it into this magnificent segmented structure.
And what's truly remarkable to me is that all of this, the bone, the ribs, the dermis of the back, and the majority of the body's skeletal muscles, it all springs from this one population of cells.
Called the paraxial mesoderm.
Right, which runs right alongside the developing neural tube and the notochord.
That's the source.
Yeah.
The somites are derived directly from these paraxial mesodermal populations. And if we look at their ultimate fate, we can summarize their massive job description into, let's say, three main categories for you.
Okay, let's break it down.
First, they create the axial skeleton. That's your vertebrae, your ribs, and your sternum.
The whole framework.
The whole framework. Second, they generate the dermis that covers your back. And third, they form virtually all the skeletal muscles of the back, the body wall, and critically, the limbs.
That's a huge organizational task for a small group of cells.
It's an enormous task.
Okay, so let's unpack this. We can't just leap to the final structure, to the adult vertebra. We have to appreciate the choreography. Our sources suggest a kind of three-part journey: a clock, followed by a split, and then a masterful reassembly.
And that roadmap is absolutely essential for navigating the molecular details. We're going to start with somitogenesis. That's the rhythmic process, the clock, as you call it, that creates the segmented units themselves.
Then the split.
Then we'll track the segmentation and patterning within each individual somitis. We'll watch it split into its two major lineages, the sclerotome and the dermomyotome.
And finally, where they end up.
Exactly. We'll follow those lineages to their ultimate destination. We'll show how the sclerotome builds the bone through this miraculous process of resegmentation, and how the dermomyotome builds all the muscle through extensive migration.
And understanding the precision here it's paramount, because the
Yeah.
developmental windows we're talking about are incredibly narrow.
Incredible.
These are events happening in the span of days, but if they go wrong, they result in lifelong structural and neurological challenges.
Absolutely. The sensitivity of this process is what makes it so fascinating from a scientific standpoint, but also so critical to clinical understanding.
Okay, let's start the clock then. Section 2, somitogenesis, the rhythmic clock of development. This is the sequential creation of those paired somites along the body axis. What makes this process so rhythmic?
The rhythm is dictated by something called the segmentation clock. This is a highly coordinated cyclic process within the tissue that comes just before the somites. It's called the presemitic mesoderm, or PSM.
So it's unsegmented tissue waiting its turn.
It is. And this clock involves the repeated cyclical expression and then the downregulation of specific regulatory genes. You can imagine it like a waveform, a pulse of gene expression.
A wave that travels down the embryo.
Exactly. And when that gene expression cycle drops below a certain threshold, boom, a new somite boundary is established.
So it's not a static line of demarcation being drawn. It's an oscillating molecular rhythm that determines when the next segment should be nipped off from the continuous tissue behind it.
You've got it.
Ugh.
And before they can be officially recognized as a somite, those cells in the PSM have to undergo a major identity shift.
What kind of shift?
They start as loosely associated mesenchymal cells. They're sort of a disorganized soup. And they have to transition into a tightly packed, organized epithelial cell ball. That's the pre-imite transformation.
And the studies highlight just how variable this timing is across species. It takes 12 hours in a chick embryo for this transformation, but only 4 hours in a mouse.
Right.
What does that difference tell us about the relationship between time, size, and segmentation?
It tells us the pace is fundamentally linked to the final product. I mean, the cycle period is species-specific. The total length of the vertebral column, the number of vertebrae, it correlates directly with how long that cycle takes.
So a faster cycle might mean more, smaller segments, or just a faster build time.
It's more about the build time and the coordination with overall growth. A faster cycle lets the animal build its segmented body plan much more quickly.
Which brings us to the human timeline, where, the pace slows down dramatically. It makes the process of segmentation a long, drawn-out affair in embryonic terms.
Oh, it's very protracted in human embryos. The sources indicate that 16 pairs of somites form between approximately day 20 and day 30 of gestation.
10 days for 16 pairs.
If you do the math, it's about 1.2 pairs per day. This slower pace means the human embryo is, well, it's vulnerable to disruptions for a much longer period than, say, a mouse embryo. It takes a full 9 to 10 days just to establish the primary segments of the trunk.
That significantly longer window of vulnerability, that really is a key takeaway.
It is.
Now, let's talk about the actual boundary creation. What specific molecular switch flips to tell one group of cells, okay, you are now somite 24, and the neighboring group, you're still just PSM?
That precise boundary formation relies heavily on signal thresholds. And the key player here is the notch signaling pathway.
Notch, again, it comes up in so many developmental processes.
It's fundamental. The mechanism is really elegant. The segmentation clock is ticking away with its oscillating gene expression. The newly defined somite boundary forms at the precise moment when the production of notch receptors, particularly in what will become the posterior or caudal part of the new somite, drops below a certain threshold.
So the cells that are about to become the back half of the new somite are actively choosing to stop listening to the surrounding signals.
Exactly.
And that allows them to sort of crystallize into a fixed structure.
That's a great way to put it. They transition from that disorganized mesenchymal tissue into the tightly packed, distinct epithelial somite. And this entire wave of somite formation moves strictly in a craniocaudal direction.
Head to tail.
Always head to tail. So the oldest, most differentiated somites are always near the head, and the newest, most disorganized tissue is always at the tail end.
This inherent rhythm, this dependence on a clean, consistent signaling drop, that seems like a major vulnerability point in development.
It is.
If the notch pathway or the genes regulating that segmentation clock are disrupted, what are the inevitable clinical outcomes?
Well, if that boundary formation is defective, the outcome is often severe. It's categorized under a group of disorders called segmentation defects. You might end up with fused vertebrae, which are known as block vertebrae.
Okay.
Or you could have missing vertebrae, or even hemivertebrae, where only half of a vertebral body forms.
And all of those lead to spinal deformities.
Major congenital spinal deformities, most commonly scoliosis, or even more severe syndromes like Klippel-Feil syndrome, which we'll definitely get into later. So that rhythm isn't just an interesting biological quirk. Its flawless execution is absolutely essential for a functional, straight spine.
So the segmentation clock ensures the somites form at the right time and in the right number. Now let's move on. Section 3. Segmentation and patterning of paraxial mesenchyme. Once that epithelial ball, the somite, is formed, it immediately faces its first identity crisis.
It's a very rapid period of cell fate determination. The epithelial somite, this uniform ball of cells you're picturing, almost immediately begins a process called ingression.
Ingression, so moving inward.
Exactly. This is where specific cells detach from that epithelial sheet and they migrate inward, changing their fundamental identity as they go.
Which cells detach first and where do they go?
The cells from the ventromedial, so that's the inner front portion, and the ventrolateral, the outer front portion of the somite epithelium, they initiate this ingression.
So the whole bottom part of the bulb.
Pretty much the whole ventral half. They move inward through the lateral aspect of the somite, and as they ingress, they undergo a crucial epithelial to mesenchymal transition, or EMT.
We've seen that before. They lose their tight organization.
They lose their tight connections, they become loosely arranged, and they form the sclerotome.
And that's the bone and cartilage precursor.
That's it. The sclerotome is everything that collapses inward to become this flexible, migratory, bone-producing tissue.
Leaving the organized shell, the dermomyotome, behind.
Precisely. The remaining epithelial sheet forms the dermomyotome, and that is destined to produce the dermis and all the skeletal musculature. This initial segregation is It's the blueprint for the entire axial structure.
But the sources reveal a fascinating detail here. Even before this ingression happens, the epithelial somite is already split down the middle.
It is?
There's a permanent segregation between the rostral or cranial half and the caudal or posterior half.
That internal segregation is the absolute key to understanding the adult spine. The ingressing sclerotome cells, they carry this identity with them. Rostral cells form the rostral sclerotome half, and caudal cells form the caudal sclerotome half.
Or they don't mix.
Crucially, as the source material stresses, these two populations never mix. They maintain their molecular and spatial identity throughout their entire migratory phase.
That fixed identity has to be enforced by something. There's a tight molecular signal stack, right? Let's talk about the primary signal that drives these sclerotome cells inward toward the midline structures, the developing neural tube and the notochord.
The primary signal governing this ventromedial migration is the sonic hedgehog pathway. Shh.
Our old friend, sonic hedgehog.
It's a potent morphogen. It's secreted by the adjacent notochord and the floor plate of the neural tube. Shh. Acts like a molecular lure, just drawing the sclerotome cells toward the midline. It's essentially telling them, come over here. You are going to form the vertebral body right here.
So shh is the magnet, but we still need to explain that permanent internal difference between the rostral and caudal halves. Why must these two halves remain so distinct, especially once they become mesenchymal and are all mixed up physically?
That distinction is paramount for the developing nervous system. It turns out the caudal portions of the sclerotome express specific molecules that are highly repulsive to migrating neural crest cells and, critically, to the developing spinal nerves.
So the caudal half is essentially a biological no-entry sign for the nerves,
That's a perfect analogy.
while the rostral half is a permissive pathway, a green light.
Functionally, yes. This permanent molecular difference ensures that the spinal nerves will only grow through the rostral half of the sclerotome. This is a non-negotiable prerequisite for the resegmentation event that happens immediately afterward. It guarantees the nerves are correctly positioned between the final vertebrae.
That is a truly elegant control system, so the boundary is defined by a notch drop, but
Yes.
the internal identity, repulsive or permissive, is set by these downstream molecular signals.
Precisely.
We're also talking about synchronization across the entire axis. There are dozens of these somites forming. What mechanism coordinates the timing across all of them?
Again, we find the notch signaling pathway is governing that synchronization, acting across the somites alongside key transcription factors, specifically T-box transcription factors. This ensures the clock ticks uniformly all the way down the axis.
But what about locking in that rostral-caudal identity difference?
For that, you need specific patterning molecules. For instance, the expression of an enzyme called RALD2, it's a retinoid synthesizing enzyme, is specifically restricted to the rostral half.
So RALTS2 isn't just some random enzyme, it's a permanent chemical flag. If you express RALTS2, you are the permissive rostral segment, regardless of which somite you came from.
Exactly right.
And you are destined to fuse with the caudal segment behind you.
It's an indelible molecular fingerprint.
Yeah.
And all of this is coordinated by external signals, too. It's not happening in a vacuum. There are constant interactions with the adjacent neural tube and the surface epidermis, involving crucial pathways like Wnt and fibroblast growth factor signaling, or FGF.
So everyone is talking to everyone else to keep the pattern straight.
The crosstalk is constant. It ensures the overall body plan development is perfectly synchronized.
Okay, let's ground this in the human timeline for our listener. When does this critical ingression and patterning phase occur in human development?
The first sclerotome cells, they begin to appear at what's called stage 14.
Yeah.
And by stage 17, the entire mesenchymal layer has fully formed. It's completely surrounding the neural tube and the notochord.
And where does it start?
The very first segments to organize are the occipital somite 1, OS1. This marks the crucial beginning of the spine, right at the base of the skull.
Which sets us up perfectly for section 4, the sclerotome, foundation of the axial skeleton. We've tracked these cells as they migrate inward. Now we follow them as they commit to becoming cartilage and bone.
The sclerotome is the bone and cartilage producing lineage. Once they become mesenchymal, these cells migrate away from that ventral somite, they multiply, and they condense to form the precursors of the vertebrae and the ribs.
This condensation isn't random, though. The sclerotome further differentiates into distinct zones based on where the cells migrate in relation to the neural tube and notochord. The source material details 4 specific subdivisions, each with a very different structural fate.
Yes, that structural definition is established very early on. First, you have the ventral sclerotome. These cells migrate most medially. They immediately surround the notochord.
So they form the core?
They form the core of the vertebral body, the centrum, and they also contribute to the annulus fibrosus, which is the outer ring of the intervertebral disc.
Okay, what's second?
Second, the dorsal sclerotome. These cells migrate dorsally, so up and around the developing neural tube. They are the architects of the back structures.
The spinous process, the bony parts you can feel on your back?
The spinous process and the dorsal parts of the vertebral arch, so the laminae and pedicles that protect the spinal cord. Third is the central sclerotome. This population sort of remains in the middle of the mass. It will contribute directly to the formation of the intervertebral disc and the ventral parts of the vertebral arch.
And finally, the lateral group.
And finally, the lateral sclerotome. These cells move furthest laterally, away from the midline, and they give rise specifically to the distal portions of the ribs.
That is incredible pre-patterning. A single epithelial somite essentially pre-patterns the entire circumference of the future vertebra and its associated rib.
It's amazing.
The cell fate is sealed before the first piece of cartilage is even laid down.
And this migration and condensation is a highly interactive process. The formation of this thin-walled, loosely arranged mesenchymal cell layer. It's not passive. It relies entirely on continuous molecular crosstalk feedback loops involving the notochord, the overlaying dermal myotome, and the surrounding ectoderm.
As they condense, they start to express the foundational materials. What signals that transition from a generic mesenchymal cell mass to the actual building material for bone?
Commitment to the chondrogenic, or cartilage-forming, lineage is confirmed when the cells forming the vertebral body begin to express type 2 collagen.
Okay.
This is the characteristic protein of cartilage matrix. It's the signal that they are now laying down the template that will later be replaced by bone through ossification.
Let's focus on the ribs for a moment. They're a massive lateral extension of the axial skeleton, but they're only prominent in the thoracic region. How do the ribs emerge, and do they also participate in that segmentation mixing?
They absolutely do. Ribs are a direct derivative of the sclerotome formed by that lateral sclerotome population.
Yeah.
And just like the vertebrae they attach to, their formation involves that fusion principle.
So it's not one sclerotome, one rib.
No. They arise through the combined expansion of the caudal half of one sclerotome and the rostral half of the next caudal pair.
So the ribs themselves are intersegmental structures. They're built from two adjacent sclerotome halves, just projected laterally around the torso.
Precisely. Their cells condense and fuse, extending laterally to form what's called the costal process. Now, this process starts in all somites, but it's only maximally developed and genetically permitted to form full ribs in the thoracic segments, so T1 through T12.
And the coordination is key.
The coordination is everything. The development of the ribs is synchronized with the development of the intercostal arteries and the associated muscles, ensuring that the vascular, muscular, and bony segmentation is perfectly aligned.
This sets the stage for what I think is the single most important and, frankly, complex concept in this entire deep dive, resegmentation. This is Section 5, development of the vertebrae and intervertebral discs. This is the structural genius that enables us to move.
Resegmentation is the fundamental concept. If you really want to understand the spine, you have to get this. If you look at an adult spine, the spinal nerve exits between two vertebrae, and the muscles span those same gaps. This functional relationship is only possible because each definitive vertebra, the final bony structure, is formed by the fusion of the caudal half of one somite and the rostral half of the somite immediately posterior to it.
Okay, let's use a clear example for our listener because this can be tricky to visualize. If we have sclerotome A and sclerotome B right next to each other, how do they combine to make one final vertebra?
Okay, sclerotome A breaks down into its rostral and caudal halves. Sclerotome B, right behind it, does the same.
Got it.
The caudal half of sclerotome A, the back half, breaks away, and it fuses with the rostral half of sclerotome B, the front half. This combination of the back of A and the front of B creates a single, functional, definitive vertebral precursor.
So what happens to the leftover bits, the front of A and the back of B?
The remaining halves, the rostral half of A and the caudal half of B, will go on to form the intervertebral disc complex that sits between the newly formed vertebrae.
Why does the body need to perform this complex mixing and matching? I mean, what is the core biological necessity? Why not just have one somite become one vertebra?
The necessity is entirely anatomical and functional. If the vertebrae were formed from single unmixed somites, the spinal nerves, which are segmented and tied to their original somite level, would have to exit directly through the center of the bony vertebral segment.
Which would be a disaster for movement.
It would completely restrict movement and potentially compress the nerve roots every time you bent or twisted. By resegmenting, the vertebral body shifts half a segment. This creates a space, the intervertebral foramen, for the nerve to exit cleanly intersegmentally between the two newly formed vertebral bodies.
It effectively decouples the skeletal segmentation from the neurological and muscular segmentation.
That's it.
It allows the nerves and muscles to operate across a moving joint rather than being trapped within a fixed segment.
Exactly. And this resegmentation works perfectly in conjunction with the molecular patterning we discussed before. Remember how the spinal nerve grows preferentially through the rostral half of the sclerotum?
It avoids the repulsive caudal half.
Right. So that nerve naturally ends up positioned in the gap created by the fusion of two half segments. It is a brilliant example of form-following function at the earliest stage of life.
Now that the main vertebral body mass is defined, let's look at the critical component that facilitates all this movement, the intervertebral disc. How is this complex, flexible structure built, and where do the materials originate?
The intervertebral disc is derived from those sclerotomal cells that don't condense to form the vertebral body itself, and it's composed of two main parts with very distinct origins.
Okay, we have the inner core, the gelatinous, shock-absorbing part, the nucleus pulposus.
That inner core is derived primarily from the remnants of the notochord. The notochord, which was that rigid axial rod that signaled all the patterning, largely regresses. But its cells persist, and they migrate with the caudal sclerotomal population.
And they become the nucleus pulposus.
They ultimately form the highly hydrated gelatinous matrix of the nucleus pulposus.
And surrounding that, you have the tough, multi-layered ring that keeps that core contained, the annulus fibrosus.
The annulus fibrosus develops from the more fibrous cells of the cranial or rostral sclerotome. These cells surround the notochord remnant and the developing nucleus pulposus, forming that dense circumferential ring.
So just like the vertebra itself, the disc is also a product of both rostral and caudal segments.
It is. The annulus from the rostral half and the nucleus pulposus from the notochord remnant associated with the caudal half.
It's astounding that this complex joint is defined so early. What is the human developmental timeline for the disc?
Our sources show that the intervertebral discs are defined quite early, by stage 17 in human embryos. But they are far from mature.
They keep developing after birth.
Oh yeah, they do not reach their adult form until long after birth. They continue to grow in size and increase their hydration up to post-fertilization week 12, and then continue to mature throughout early childhood.
Now let's transition from cartilage to bone, the ossification of the vertebral body. How does that cartilaginous template become the hard, mineralized structure we rely on?
The process is endochondral ossification, the conversion of a cartilage model into bone. The vertebral body is fully cartilaginous by stage 17, so roughly 7 weeks gestation. Ossification begins from 3 distinct primary centers for a typical vertebra.
Can you detail those 3 centers for us?
Sure. For a typical thoracic vertebra, you have one central primary ossification center for the body. That's called the centrum.
Okay.
Then you have 2 lateral primary centers, one for each side of the pedicles and laminae, which together form the neural arch that protects the spinal cord.
When do these centers appear, and how does the bone growth spread?
The primary centers first appear at stage 23, so around 8 weeks gestation. They begin in the lower cervical and upper thoracic regions first, and then the process spreads both cranially up the neck and caudally down the back.
And the fusion is a very protracted postnatal event.
That's vital for understanding pediatric growth. The 2 halves of the neural arch, which surround the spinal cord, they fuse posteriorly between 1 and 6 years of age.
Wow, that late.
And the centrum, the body, then fuses with those arches between 3 and 6 years. Even later, you have secondary ossification centers, which form the very tips of the spinous and transverse processes, and they don't appear until much, much later, often not until puberty.
So some parts of the spine are not fully fused until your mid-20s.
That's right. The maturation of the spine literally spans decades.
The final takeaway from this section, then, has to be the anatomical genius of resegmentation, how it dictates nerve placement.
Absolutely. The system is designed to guide the spinal nerve through the non-repulsive rostral half of the sclerotome. When that rostral half fuses with the caudal half of the segment in front of it, the definitive vertebra is formed, and the nerve is perfectly framed to exit the vertebral column laterally, precisely where it needs to be to innervate its corresponding body segment. It is segmentation allowing for movement.
That structural framework is complete. Now we have to move to the engine of that movement, the musculature. Section 6 focuses on the myotome, building the body's musculature. We left off with the dermomyotome, that remaining epithelial shell after the sclerotome ingressed.
Right. The dermomyotome is the source of two crucial lineages, the dermis covering the back and virtually all the skeletal muscle in the body, with the exception of some specialized head and pharyngeal arch muscles.
How does the myotome, the muscle precursor, mass form from this epithelial sheet?
Cells detach from two specific edges, the dorsomedial lip, which is the upper inner edge, and the ventrolateral lip, the lower outer edge of the dermomyotome. They ingress into the space between the neural tube and the ectoderm. This process of forming muscle cells, or myogenesis, is highly conserved across all vertebrates.
And what are the key molecular flags that signal this commitment to muscle fate? What says, you're a muscle cell now?
Myogenesis is clearly marked by the expression of master regulatory transcription factors. The big two are MyoD and My5.
MyoD and My5.
These factors initiate the entire cascade that commits the cell to a muscle lineage. If these genes are expressed, that cell is destined to become a muscle fiber.
This brings us to another critical organizational split, maybe the most critical for understanding functional anatomy, the apaxial versus hypaxial division.
This split determines the location and the innervation of every major skeletal muscle in your trunk and limbs. It is dictated entirely by where the myotome cells migrate after they ingress.
Okay, let's start with apaxial.
The apaxial muscles are the dorsal muscles. They're derived from that dorsomedial lip. These cells remain close to the neural tube and the dorsal aspect of the spine.
They don't travel far.
They stay home. They form the deep intrinsic muscles of the back, the erector spinae group, the deep stabilizers, and they're innervated exclusively by the dorsal rami of the spinal nerves. Their job is stabilization and extension of the spine.
So if you're trying to stand up straight or arch your back, you're using your apaxial muscles, and they haven't had to travel far from their origin point at all.
Precisely. Conversely, you have the hypaxial muscles.
The ventral ones.
The ventral ones, they're derived from the ventrolateral lip, and these are the highly migratory cells. These cells travel extensively into the lateral body wall and, importantly, into the developing limbs.
And they form most of the body's muscle mass.
The vast majority. The abdominal wall, the intercostals, and all limb muscles. They are innervated by the ventral rami of the spinal nerves.
The functional distinction is remarkable. Apaxial muscles stabilize the axis. Hypaxial muscles move the periphery and manipulate the body cavity.
That's a perfect summary.
The extent of this hypaxial migration, especially coming from the cervical region, is just astounding when you dig into it.
It's one of the most stunning examples of developmental relocation. The hypaxial muscles of the thoracic region, they form the intercostal muscles and the rectus and oblique abdominal muscles. That's fairly straightforward. But the precursors from the cervical somites, the neck area, they undertake a colossal journey.
They migrate all the way down to form the infraoid strap muscles in the neck and, most crucially, the diaphragm.
Yes. The diaphragm, the primary muscle of respiration, starts its life as a cervical precursor cell. It migrates all the way down the torso,
dragging its nerve supply with it.
Dragging its innervation, the phrenic nerve, which is a ventral ramus derivative of the cervical plexus, all the way down to the base of the rib cage. This is why the nerve pathway is so long and indirect. It originates in your neck to control a muscle in your abdomen.
When you realize the diaphragm's muscle cells began as a cluster of cells thousands of units away from their final destination, carrying their nerve supply along for the ride, it completely changes how you view respiratory anatomy.
It demonstrates the primacy of the developmental plan. And the hypaxial contribution is equally robust caudally, at the tail end.
What does it form down there?
It forms the lower abdominal muscles like the transversus abdominis and the specialized musculature of the pelvic floor, including the anal and urethral sphincters. The hypaxial compartment literally builds the structural periphery, the essential functional walls of the body cavities.
Let's briefly return to the dermo part of the dermomyotome. The sources note a sharp boundary here. Where does the somite-derived dermis end?
The contribution is strictly limited. It only forms the dermis covering the back. The boundary is very sharp. The dermis covering the limbs and the ventral body wall is derived from a different source, the somatopluric mesenchyme of the lateral plate.
That makes perfect sense, functionally. This is why our dermatomes, the segments of skin innervated by a single spinal nerve, correlate so precisely with the underlying deep muscle segments, the myotomes, created by that same somite.
It's a beautifully integrated system. The skin on your back is wired to the deep muscles underneath it because they both came from the same original
A
segment.
perfect developmental package.
It is. And we should also acknowledge the somites often overlook derivatives. The early ventral half of the somite also gives rise to smooth muscle cells around the somites and in the tunica media of the descending aorta. It even produces angioblasts, which are the precursors to the endothelium lining our blood vessels.
So, the somite isn't just the axial architect. It's a temporary multi-purpose factory providing skeletal, muscular, dermal, and even vascular elements for the central body axis.
The truly versatile and temporary organ.
Okay, now we shift our focus to Section 7. Regional variation in vertebral development. The structure we've described, the formation of a single vertebra, is the blueprint, but the final product is clearly not uniform. We have very specific cervical, thoracic, lumbar, and sacral structures. How does the generic somite template differentiate into these regional forms?
This regional identity is dictated by complex regulatory genes, primarily the Hox genes.
The master body plan genes.
Exactly.
Yeah.
These genes are expressed in overlapping domains down the anterior-posterior axis, and they act like molecular zip codes. They tell the somite what type of segment it must become: neck, rib-bearing, or lower back.
Let's start at the very top, the most complex transitional zone, the occipitocervical junction. This is where the spine meets the base of the skull.
This junction involves the first four occipital somites, OS1 to OS4, being co-opted into forming elements of the skull base rather than full cervical vertebrae. The joint forms between the occipital bone and cervical vertebra 1, C1, also known as the atlas.
What happens to the very first sclerotome, sclerotome 1?
Its fate is highly specialized. The most caudal portion of sclerotome 1 usually contributes directly to the base of the skull. It specifically forms part of the exoccipital bone. But more remarkably, the centrum, the body precursor of sclerotome 1, actually separates.
It detaches.
It detaches and descends to fuse with C2, the axis, later on in development.
And that separated centrum forms the dens, or the odontoid process, which C1 rotates around.
Exactly.
So the pivot point for your head rotation comes from the very first somite, but it's permanently attached to the second cervical vertebra.
That is the rearrangement. And C1, the atlas itself, is formed primarily by the central portion of sclerotome 2, which forms its ventral arch. Its dorsal arches derive from the dorsal parts of sclerotomes 2 and 3.
It's a real mix and match zone.
It's a notorious zone for developmental abnormality because it involves the rearrangement, separation, and eventual fusion of three different somites into the two highly specialized rotational vertebrae, C1 and C2.
Moving slightly further down, let's examine the transitional vertebra bridging the neck and the thorax. Cervical vertebra 7, C7. This one often behaves differently than its neighbors.
C7 is the perfect example of a transitional segment under strong Hox gene regulation. Its transformation into a thoracic-like vertebra involves its laminae becoming more horizontal and its spinous process becoming markedly longer and more caudally directed. But the defining feature is the propensity of its costal process to develop into a rib.
The appearance of cervical ribs.
Indeed. C7 possesses long transverse processes that frequently develop full or rudimentary ribs. This is often linked to a slight anterior shift in the Hox gene expression boundary.
And this can cause problems.
While many people with cervical ribs are asymptomatic, if the rib is large enough, it can impinge on the brachial plexus or the subclavian artery, leading to neurological or vascular symptoms, a condition known as thoracic outlet syndrome.
This link to Hox gene boundary shifts is critical. If the Hox code for thoracic leaks into the C7 segment, you get a rib where there shouldn't be one. And the same boundary problem can lead to more severe congenital fusions, like Klippel-Feil syndrome.
That is the mechanism behind Klippel-Feil. The abnormal segmentation and fusion are often traced back to disruptions in the signaling cascades, and thus the Hox code governing that occipitocervical transitional zone, resulting in a short, immobile neck.
Once we reach the thoracic region, T1 to T12, we hit the structural maximum. This is the domain of full ribs.
The thoracic vertebrae are defined by maximum lateral sclerotome growth. The ribs develop via costovertebral joints, which are synovial joints, allowing for movement during respiration, connecting the proximal rib to the vertebral body and the transverse processes.
And here we see the differentiation of true, false, and floating ribs based on their attachment to the sternum.
Right. Ribs 1 through 7 are the true ribs. They attach directly to the sternum via their own costal cartilage. Ribs 8 through 10 are the false ribs. They attach indirectly, connecting to the cartilage of the rib above.
And then the floaters.
And ribs 11 and 12 are the floating ribs. They're short and they don't connect to the sternum at all. This differentiation reflects the complex structural needs of the body wall, balancing protection and mobility.
Finally, let's travel down to the robust structures of the lumbar and sacral regions.
In the lumbar region, the vertebrae are defined by their immense size and thickness, which is necessary for weight bearing. Here, the costal process does not form a rib. Instead, it remains incorporated into the large, prominent transverse processes of the lumbar vertebrae. The discs here are exceptionally thick.
And the sacrum, the fused foundation at the bottom.
The sacrum is composed of 5 fused segments, S1 through S5, and they're typically narrower than the segments above. The fusion process is one of the longest in the body. While S1 through S3 may show signs of fusion at birth, the complete fusion of all sacral segments doesn't finalize until young adulthood, typically around 25 years of age.
We mentioned variation earlier, the incidence of having 6 lumbar vertebrae, for instance.
Variations are quite common. Sometimes up to 10% of the population exhibit variations like 6 lumbar vertebrae, which is called lumbarization, or 5 of the transitional sacral vertebra, called sacralization.
And it's usually not a problem.
As long as these variations don't affect neurological function, they're usually asymptomatic. However, if the variation disrupts the alignment or the movement potential at a major joint, like the lumbosacral junction, it can lead to chronic biomechanical issues.
We've built the back piece by piece. Now we move to Section 8, clinical correlates and final synthesis. What happens when these incredibly precise developmental pathways fail?
The precise timing of segmentation, migration, and fusion is, as we've said, a developmental high-wire act, and disruption leads to major congenital defects. The most well-known category is spina bifida.
Spina bifida is fundamentally a failure of the fusion of the vertebral neural arch, most frequently in the lumbar and sacral regions. But what is the embryological root cause of this failure?
The failure is twofold. It begins with the neural tube defect. The neural plaque fails to sink properly into the embryo and cover the cord.
So the problem starts with the nervous system.
It does. And this means the overlying dorsal sclerotome cells, the population destined to form the dorsal arch and spinous process, do not receive the correct signals to migrate and condense over the midline. If the arch doesn't form, the spinal cord is left exposed.
Let's detail the spectrum of severity, starting with the mildest, spina bifida occulta.
Occulta, meaning hidden, is the simplest failure of the arch fusion, often involving just 1 or 2 spinous processes. It's typically covered by skin.
So you can't see it from the outside.
You might see a sign. It may be noted by a tuft of hair, a small dimple, or a patch of fat, a lipoma, over the area. It is usually asymptomatic because the neural tissue itself is not involved.
But the more severe categories, known as spina bifida cystica, involve protrusion.
Yes, cystica involves a visible, fluid-filled sac. Meningocele is the lesser severity. It involves the protrusion of the meninges, the protective membranes, through the bony defect.
But the spinal cord is okay.
The cord is in the right place. But the most clinically significant and common severe form is meningomyelocele.
And that's where the cord itself is in the sac.
That's where the cystic sac contains both the meninges and the neural tissue itself, the spinal cord and/or nerve roots. This exposure leads to permanent neurological deficits below the level of the defect.
And the severity of these forms really underscores the importance of nutritional intervention, specifically folic acid supplementation, which has to happen extremely early.
Absolutely. The window for prevention is incredibly narrow, highlighting the sensitivity of these initial stage 15 events.
Let's discuss 2 related caudal defects that are also rooted in faulty development: tethering of the cord and diastematomyelia.
Tethering occurs when the spinal cord is abnormally fixed or attached to structures at the caudal end, often to the dura mater or the skin. This relates to the phenomenon of differential growth.
Meaning the bones grow faster than the cord.
Right. The vertebral column grows longer and faster than the spinal cord. Normally, this differential growth causes the spinal cord to terminate higher in the vertebral column, around L1 or L2 in adults.
So if the cord is tethered, it can't ascend relative to the column, and it gets stretched as the child grows.
Precisely. This stretching results in ischemia and dysfunction, leading to progressive sensory, motor, and bladder problems.
And what is diastematomyelia?
Diastematomyelia is a condition where the spinal cord is split longitudinally into 2 distinct hemichords, often associated with a bony or fibrous spur that drives the split. This condition is also typically accompanied by severe vertebral defects in the same region.
We've covered the failures. Let's quickly review the postnatal process once more. The ossification timeline continues for decades.
That process is long and directional. Primary ossification centers proceed craniocaudally, starting early C1 around week 8 and moving down to the sacrum around week 22. Secondary centers only appear much later, around puberty, providing the growth potential for the final shape of the processes and apophyses.
The lesson here seems to be that most structural variation is tolerated, but anything that disrupts the neurological or joint alignment is problematic.
That is the synthesis. Variation in segment number or accessory ossification centers is common and usually benign. However, if the timing of fusion is disrupted, as seen in conditions like achondroplasia where the growth plates of the vertebrae are affected, or if the alignment in the highly mobile atlantoaxial or lumbosacral transitional zones is faulty, then the consequences for biomechanics and neurological function are severe.
We have completed the full arc of the somite's life, from rhythmic inception to final segmented structure. Let's synthesize the 3 key insights that should stick with you, the listener, from this deep dive.
First and foremost, you have to remember the genius of resegmentation.
Right.
The spine is not built in whole segments, but through the fusion of the caudal half of one somite with the rostral half of the next. This mixing is the non-negotiable mechanism that allows the spinal nerves to exit properly between the vertebrae.
Enabling segmented and flexible movement.
Across the whole axial structure. It's everything.
Secondly, the molecular control dictates destiny. The entire developmental structure is a coordinated signaling environment.
It is. The clock, the notch pathway, tells the somite when to form. Molecules like sonic hedgehog draw the cells to the midline, and internal markers like REL2 set the rostral caudal identity, a molecular fingerprint established before the cell ever reaches its final bony home.
And
thirdly, recognize the profound distinction between the musculature, epaxial versus hypaxial. That initial developmental split dictates the entire functional landscape of your body. It separates the deep intrinsic stabilizers of your back, innervated by the dorsal rami.
From the vast migratory muscles that control your limbs, your abdomen, and crucial organs like the diaphragm, all innervated by the ventral rami.
You've got it.
What a journey of transformation. From a simple segmented ball of epithelial cells, we get the entire structural axis of the body intricately linked to the nervous system. It really is a feat of biological engineering.
It truly is. And this level of precision leaves us with a provocative question to consider as you wrap up this deep dive.
Give us the final thought.
If the difference between a muscle that never moves more than a millimeter from the spine and a muscle like the diaphragm, which travels the entire length of the torso, originates in the subtle developmental paths taken by a somite cell thousands of cells wide, what subtle yet powerful influences might these precise embryonic migration patterns have on individual biomechanics, flexibility, and even vulnerability to musculoskeletal injury that we still frequently attribute only to fixed genetics?
That suggests that the environmental and nutritional status during those first few days of segmentation might encode subtle differences in our structural resilience that we haven't fully recognized. It's a fascinating challenge to the purely genetic view of body structure.
It encourages us to look deeper into the implications of that rhythmic, vulnerable segmentation clock and how those foundational moments truly shape our functional lives.
Absolutely fascinating material. Thank you for sharing your source material and letting us guide you through this deep dive into the origins of the human axial skeleton.
Always a pleasure.
We'll catch you next time for another deep dive.